Welcome to the deep dive. We're jumping straight into some really fascinating science today, a totally fresh angle on tackling cancer.
Yeah, and it involves something you might not immediately think of, fat cells.
Exactly. So our goal here is to unpack this new approach. It's called adipose manipulation transplantation, or AMT. We want to understand how it could, well, maybe shift how we fight this disease.
Right. We're looking at some compelling research, really detailing how scientists have engineered fat cells, basically making them super competitors for nutrients.
And potentially starving tumors.
That's the idea, suppressing tumor growth. We'll dig into their experiments, everything from cell cultures, mouse models, even human tissue work.
Okay, so the core concept, and this is where it gets really interesting, tumors are just incredibly hungry for energy. They need massive amounts of fuel.
Constantly.
So the researchers asked, what if we introduce something even better at, like, grabbing those nutrients right near the tumor?
Almost creating a local metabolic sink that just pulls away everything the cancer needs.
Yeah, like a nutrient vacuum cleaner.
That's a good way to think about it. And to really get why this strategy might work, we first need to talk about the tumor itself, its environment.
Okay. It's not just the cancer cells, right?
No, not at all. The tumor microenvironment, it's complex. You got other cells, structural bits.
And the conditions are tough in there.
Often, yeah. Low oxygen, that's hypoxia and scarce nutrients. It can be a really challenging place to live.
But cancer cells manage it.
They do more than manage. They adapt. They're metabolically flexible, meaning they can actually reprogram how they use fuel to survive, even thrive in those conditions.
So they become dependent on certain ways of getting energy, like maybe they rely heavily on sugar, glucose, or specific fats.
Precisely. And that reliance is a potential weakness. It's something current therapies try to hit.
How so?
Well, you have drugs targeting enzymes like hexokinase 2, which is key for using glucose, or proteins that help cancer cells absorb fats, lipids.
So the aim is often to block those specific fuel lines.
That's a major strategy, yes. People have even looked at activating brown fat naturally in the body.
Brown fat, the kind that burns energy.
Yeah, because it ramps up glucose and fat metabolism, potentially creating competition. But the problem is, how do you activate it effectively in a patient?
You mean like making people cold?
Exactly. And doing that consistently, long term, well, it's really challenging. Maybe not feasible for many patients.
Right. Doesn't sound very practical.
Yeah.
So how does this new AMT approach get around that?
So the core idea with AMT is, instead of a whole body approach, what if we engineer fat cells themselves to be these metabolic powerhouses, cells that can locally snatch up nutrients right there next to the tumor?
Targeted intervention makes sense. How did they engineer them?
They used CRISPR activation. CRISPRa.
Okay, I've heard of CRISPR for gene editing, cutting genes, but CRISPRa.
Right, so CRISPR is different. It doesn't cut. Think of it more like a precision volume knob for genes. It turns up their activity.
Got it. So they're boosting certain genes in these fat cells. Which ones?
They focused on genes critical for brown fat, remember, the energy-burning kind.
Yeah.
The goal was to give regular white fat cells some of those key brown fat characteristics, making them more metabolically active.
So trying to brown the white fat cells, basically. Why specifically brown fat genes?
Because brown fat is really good at dissipating energy. It does this through something called non-shivering thermogenesis.
Burning fuel for heat without shivering.
Exactly. And that process demands a lot of glucose and fatty acids. So by turning up the brown fat genes in these adipocytes, that's the technical term for fat cells, they aim to create cells with a much higher appetite for fuel.
Okay, so which specific genes did they crank up with CRISPR?
Three main ones: UCP1, PPARGC1A, and PRDM16. They're all known master regulators of brown fat development and function.
And how did they get the CRISPR machinery into the cells?
They use viruses, adeno-associated viruses, AAVs. Think of them like tiny delivery trucks. They delivered the CRISPR components into human white pre-adipocytes, the precursor cells. Then they let them mature into adipocytes in the lab.
And did it work? Did they actually boost those target genes?
Yes, significantly. They checked using qPCR, which measures gene activity, and they saw big increases in UCP1, PPRGC1A, and PRDM16 expression compared to controls.
Okay, step one, successful.
Yeah.
But did the cells actually start acting like brown fat? Functionally?
Good question. They looked at other brown fat marker genes and saw increases there too, like TFEM, DIO2, CPT1B, mostly. Boosting PRDM16 didn't seem to upregulate CPT1B and another one, NRF1, in their setup.
Interesting nuance.
Huh?
But overall, the genetic profile shifted towards brown fat.
It looked like it, yeah. Then they measured actual energy use
Ah.
using something called a seahorse assay. It measures how much oxygen cells consume.
Yeah.
The engineered adipocytes used more oxygen overall, especially the UCP1 boosted cells. They also showed more uncoupled respiration that's key for brown fat's heat production and a higher maximum breathing capacity, essentially.
So they were working harder metabolically, burning more fuel.
Exactly. And not just oxygen. They took up more glucose too, both normally and when stimulated with insulin. Plus, they showed increased fatty acid breakdown and uptake.
Wow, so they really did manage to engineer these white fat cells to be much more metabolically active, more brown fat-like.
Seems so. They successfully browned them in the lab.
Okay, so the crucial test. How did these souped-up fat cells do against actual cancer cells?
Right. Next step, put them together. They did co-culture experiments, grew the engineered adipocytes in the same dish with various cancer cell lines.
What kinds of cancer?
Breast, colon, pancreatic, prostate, a range.
And how did they track the cancer cells' growth?
They used assays measuring cell viability, basically, if they're alive, and proliferation, how fast they're dividing. Luminescence, BRDU incorporation, standard methods.
What happened?
Pretty striking results. The adipocytes with boosted UCT1, PPRGC1A, or PRDM16, significantly slowed down the growth and division of multiple cancer lines.
Multiple lines, so it wasn't just one specific cancer type.
No, it seemed quite broad, which is promising.
What was going on at the metabolic level between the cells? Did they check that?
They did. They saw the cancer cells took up less glucose and had lower glycolytic activity, that's glucose breakdown, when grown with the engineered fat cells. And they also saw the cancer cells weren't taking in as much external fat.
So it really looks like the engineered fat cells were outcompeting them for fuel, winning the nutrient battle.
That's the strong hypothesis, yeah. They even saw the cancer cells had fewer nutrient transporters on their surface, like GLUT4 for glucose, CD36 for fats, and lower levels of metabolic enzymes.
Like their supply lines were being choked off.
Exactly.
Okay, so in a dish, it works. Starves the cancer cells. But, you know, a living body is way more complex. What about animal models?
The essential next step. They moved to in vivo testing. First, they created adipose organoids.
Organoids, like mini organs.
Sort of. 3D clusters of the human preadipocytes, which they then engineered with CRISPRa. These mimic actual fat tissue better than just cells in a flat layer.
Okay, mini fat tissues, engineered. Then what?
Then they implanted these engineered organoids into immune-deficient mice right next to human cancer cells that they had also implanted. These are called xenografts.
And watch the tumors grow?
Exactly. They tracked tumor size over several weeks.
And the results.
Tumors growing next to the UCP1 CRISPR organoids were significantly smaller. We're talking over 50% reduction in volume compared to controls.
Wow, over 50%. That's huge. Did they look inside those smaller tumors?
Yes. Gene analysis showed lower levels of MKI67, a marker for cell division, so less proliferation.
Okay.
And they used staining techniques, immunofluorescence, to actually see proteins in the tumor slices. They found less Ki67, another proliferation marker,
Right.
less CA9, which indicates hypoxia, low oxygen, and less CD31, a marker for angiogenesis, the growth of new blood vessels tumors need.
So slower growth, less oxygen stress, fewer blood vessels.
Yeah.
It's like hitting the tumor on multiple fronts.
It seems that way. And when they checked nutrient levels,
What did they find?
the engineered fat organoids nearby had higher glucose levels, but the tumors next to them had lower glucose and lower levels of the molecules made during glucose breakdown. Same pattern for fatty acids, lower in the tumors near the engineered fat.
Really strong evidence for that nutrient competition idea playing out in the living system.
Very compelling, yes. They also did RNA sequencing on the tumors, looking at all the genes being expressed.
What did that show?
In mice on a standard diet, the tumors near the engineered adipocytes had widespread gene expression changes. Lots of metabolic genes and cancer growth genes were downregulated, turned down.
Okay.
But interestingly, some tumor suppressor genes were upregulated, turned up, along with genes involved in stress response and apoptosis-programmed cell death.
So the cancer cells weren't just starved, their whole genetic program was shifting towards being less aggressive, maybe even self-destructing.
That seems to be the implication, yes.
Yeah.
It's a complex response.
You mentioned standard diet. Did diet matter?
Hugely important finding. Yes, it mattered a lot.
How so?
The tumor suppression was strongest in mice on that standard diet. When they fed mice a high-fat diet or gave them high-glucose water, the effect was much weaker.
So if there's just tons of fuel everywhere in the body,
Then the local competition created by the engineered fat cells doesn't have as much impact. The cancer cells can still get nutrients from the general circulation.
makes sense. It highlights that the local environment matters, but so does the whole system. Did they try this in mouse models where cancer develops more naturally?
They did genetic models for pancreatic and breast cancer.
Oh,
In the pancreatic model, putting UQ1 CRISPRa mouse adipose organoids near the pancreas slowed tumor growth and reduced hypoxia.
okay.
In the breast cancer model, putting engineered organoids near the mammary glands, or even far away on the mouse's back, still reduced tumor mass and volume.
Even far away. That's interesting. Suggests maybe a systemic effect too,
Right.
or just very potent local competition draining resources.
Could be either, or both. The gene expression changes in those models also pointed towards nutrient competition being key.
So this looks promising across different cancers, different models. What about getting closer to human application? Any steps there?
Several key steps. They used tumor organoids derived directly from human breast cancer patients.
Patient tissue.
And when they co-cultured these with the engineered adipocytes, they saw significant suppression of cancer progression and proliferation. That's a really crucial link.
Showing it works on actual human cancer, not just cell lines. What about prevention? Could this be used preventatively?
Fascinatingly, they tested it on breast organoids from people with BRCA mutations, high-risk individuals.
Yeah.
The engineered adipocytes suppressed cell proliferation in those high-risk organoids too. It just hints at a potential preventative role, maybe?
Wow, that's unexpected. Now, for clinical use, you'd want control, right? Can they turn these engineered cells on and off?
Yes, they developed an inducible system using tetracycline, basically a drug trigger.
So you give the drug, UCP1 turns on, you stop the drug, it turns off.
Exactly. It allows for external control, which is important for safety and managing effects. And they also worked on delivery.
How would you get these cells where they need to go and keep them there?
They created a cell scaffold platform, a biodegradable scaffold, like a tiny sponge with little wells to hold the engineered adipose organoids.
And they implanted that next to tumors.
Yes, in mice. It also reduced tumor growth and metabolic activity. And a key feature is that this scaffold could potentially be removed or replaced later.
That adds a layer of safety and control, being able to take them out if needed. Did they explore targeting other metabolic weaknesses besides just general glucose and fat?
They did, showcasing the platform's flexibility. Pancreatic cancer, for instance, can become dependent on a nutrient called uridine, especially when glucose is low.
Okay.
So they engineered adipocytes to express more UPK1, an enzyme that breaks down uridine. These cells would suck up uridine and convert it to lactate.
And did that hurt the pancreatic cancer cells?
Yes, when grown together in low glucose conditions, it significantly suppressed the cancer cell growth. And if they added extra uridine back,
The suppression stopped.
exactly, it reversed the effect, confirming it was uridine depletion. This effect was less pronounced when plenty of glucose was around, again showing context matters.
So this AMT approach seems really adaptable. You could potentially tailor it to hit specific metabolic weak spots in different cancers.
That's one of the most exciting aspects, I think, the potential for personalization, designing the fat cells to match the tumor's specific metabolic profile.
So thinking bigger picture now, what are the implications? Where could this go?
Well, AMT could represent a whole new class of cell-based therapy, maybe like SAR or T therapy, but using fat cells.
Which are easier to get, maybe.
Potentially. Adipose tissue is pretty accessible. Liposuction, fat grafting, these are common procedures already.
Any other advantages over using immune cells like T cells?
Possibly lower immunogenicity. Fat cells might provoke less of an immune rejection response than T cells, which could make developing off-the-shelf therapies easier therapies you don't have to make individually for every patient.
An off-the-shelf engineered fat cell therapy. That's quite a thought. What about downsides? Like cachexia, the wasting syndrome in cancer. Could these cells make that worse by burning too many calories?
That's a critical concern. But the inducible system, the ability to turn the cells off, or the removable scaffold, those offer ways to potentially manage such side effects. If the effect becomes too strong systemically, you could dial it back or remove the source.
So future research would likely involve refining that control, exploring other metabolic genes to target maybe better delivery methods.
Absolutely. Optimizing the engineering, the delivery, understanding long-term effects, safety. There's still a lot to investigate, but the potential is clearly there.
Okay, this has been really eye-opening. Let's try to wrap up the main takeaways.
Sure. So fundamentally, this research shows that engineered fat cells, these AMT cells, can compete effectively with tumors for nutrients.
Mm-hmm.
And this competition significantly suppresses tumor growth in a variety of lab and animal models. It really opens up a novel therapeutic avenue.
With possibilities for personalization, control.
Exactly.
And for me, the real aha moment is just the concept itself. Using supermetabolic fat cells to starve cancer, it flips the script on how we usually view fat and disease,
Right.
repurposing it as a weapon.
It's a definite paradigm shift, yeah.
Yeah.
A really clever biological approach.
So for you listening, this offers a glimpse into the kind of innovative thinking that could lead to, hopefully, more targeted, maybe less toxic cancer treatments down the road. Manipulating metabolism at this cell level, it just shows the power and complexity of biological engineering.
And it leaves us with a provocative thought, doesn't it? We understand there are relates between obesity, metabolic issues, and cancer risk. Could an approach like AMT be refined even further?
How do you mean?
Could it not only target the tumor's nutrients, but maybe also help address some of the patient's underlying metabolic problems simultaneously? And thinking even broader, fat cells are just one type of cell. What other helper cells in our bodies could we potentially engineer to fight diseases in new ways?
That's a great question to ponder. Lots of possibilities there.
Definitely food for thought.
Indeed. Well, thanks for joining us for this deep dive. Until next time, keep exploring.